All right. Hello viewers, welcome back. This is Dr. Abid. Viewers, in today's lecture, we will be discussing about collection of the semen to the cryopreservation technique to store the semen and preserve its quality and quantity. So let's start our lecture. First of all, what is semen? Semen is defined as a bodily fluid that is secreted by the male genital organs. It consists on two parts. Number one, viable part, that is sperm or the spermatozoa, and another part that is considered as fluid, which contains citric acid, free amino acids, fructose, phosphorycholine, prostaglandins, potassium, zinc, and other enzymes which are essential to maintain the viability of the sperms and facilitate the fertilization in later stages. So here is the animal which shows that semen is produced from the testes and then converted mature stages, later stages. These are matured into various uh, body organs of the male animals and then transported to the female reproductive tract via the penis. So this is the just diagrammatic representation. And then after collection, the semen is stored in various straws to extend its quality and then stored it at the later stages. So in our today's lecture, we will be discussing on these, focusing especially on these three parameters from the collection and then distribution into the straws of various volumes like 0.25 to 0.5 volume straws comes, and then storage into the containers. So shift in temperature from the collection to the preservation and insemination. Body temperature of the male animals from where the sperms are produced are just like testes or scrotum. Temperature is below 4 to 5 degrees centigrade than other body parts. And then semen is collected into a collection tube, like through the artificial vagina or any other means like semen collection methods. Others includes the massage method, electroejaculation method, and then it is moved to the room temperature in the water bath and then cooling chamber or cabinet to liquid nitrogen, which has the temperature to round about minus 196 degree Celsius. And then after, at the time of insemination, straws are firstly thawed in a room temperature or more probably around the 35 degree centigrade. So what is the process of collection to the storage and insemination? First of all, semen is collected from the male either via the artificial vagina means or through the direct collection from the female reproductive tract or electroejaculation methods. And then after collection, quality of the semen is evaluated in the laboratory. Various parameters including its physical, morphological, chemical, and motility is evaluated in the laboratory, and then if needed, dilution and extension is done to increase the quantity of sperm, which is the main purpose for the collection of the semen, in which the male, just like a mature or adult male, produces in billions of the billions of the sperms in a just one ejaculate. So we promote or increase the quantity of the semen by extending by using various extenders. So later on, it is stored into straws. After conversion into the straws, these are inseminated into the female animals for fertilization. So this is a roundabout. An ejaculate considered normal if contains at least So as mentioned below, before that, what are the properties of a semen or the ejaculate which is collected? It should be considered as a normal. So 60% of the spermatozoa with correct progression are considered as a normal in a one ejaculate, and 80% of the spermatozoa without any of the morphological changes, just like in changes in the tail, head or body, may cause the uh, inappropriate results. And 500,000 spermatozoa in 1 millimeter cube is essential to be considered as a normal semen for further processing. So what are the main factors which influence the fertility of the animals? Breed influence the fertility rate, breeding conditions in which the animals is to be bred, methods of the fertility, methods of the breeding, just like outbreeding or inbreeding, and method of bull 2 and 7 years of the age are the most fertile, and older ones produces lower quantity of the sperm concentration, and collection from mature bull twice a week is essential, and more than that may results in the reduced viability or the such a not satisfactory quality of the semen, and collection from up to 2 years every 5 to 7 days. Now what are the parameters for the selection of bulls to ensure? That is, physical fitness parameters are essential to identify the sire bulls for proper breeding purpose and from where the from whom the semen is to be collected. Condition of the genital organs is that normal or size of the scrotum, etc., and quality of the semen and sexual behavior of bulls are uh, considered while selecting the appropriate or desired bull for further breeding purpose. So what are the physical features of the breeding bulls? The physical fitness of bull is evaluated in the standing position and in motion on hard ground. Special attention should be paid to the position of thoracic limbs, pelvic limbs and the hoof belt, which will be shown in our next slides. Pelvic limbs are of basic importance for copulation efficiency because during mounting they take over the weight of the bull. Physical fitness is also necessary to evaluate. An analysis of the jump on dummy female or teaser is therefore performed. So here is the diagrammatic representation of physical fitness bulls to be selected for uh, breeding purpose or as a sire for your flock. So this is the correct position of their hind limbs and their lateral view and posterior view, and these are the bow-legged and cow-hocked. These are not preferred. So, how we will evaluate the male genital organs? First, we should visualize or evaluate visually, and then tactile method, and after that rectal. These are three parameters which needs to be evaluated. Male genital organs in bulls can be divided in two basic groups. Just one, internal genital organs and external genital organs. So we are not going to discuss in detail these parameters. And then sexual behavior is also checked. In the behavior assessment, two features are defined: the sex drive or libido or the ability to copulate. The evaluation of the sex drive is performed at the moment of collecting the semen, and in the evaluation of sex drive, the reaction time of the bull to the teaser or dummy cow is considered. So a 6-degree scale for sex drive, which will be discussed in our later lectures in detail. A well-balanced nutrition is important for optimal sexual activity. Excessive fatness affects negatively the liveliness of the sperms. Protein deficiency decreases sexual activity and the quality of the sperm. Disorders in the male reproductive may also be caused by vitamin deficiency, especially vitamin A, E, B groups, and lack of the microelements like zinc, selenium, manganese, and iodine. So, in the morning before the collection of semen, the bull does not receive food because a full animal has lower sex drive. So, here it is also represented that semen collection should be done at early in the morning. The collection of semen is performed in a specially prepared place, and then the place must meet appropriate sanitary and health conditions as it is considered as a laboratory room. And then the floor of the room should be flexible and anti-slip to prevent the slippery of the animals, which may result in the breakage or fracture of the animals. Initially the bull makes a few rounds in collection room and is allowed to sniff around and to jump over other bulls and to perform empty jumps on a dummy cow. A well-aroused male with distinct sexual impulses is brought to the dummy cow or the teaser in order to produce semen. Then semen is collected in an artificial vagina composed of glass container for semen with a water jacket and a volume scale, thin flexible latex sleeve, latex cone joining the end of the vagina with collection tube, rubber cylindrical casing with a valve for pouring water and blowing air, bag, a thermometer protector, and mechanical container. So these are the very necessary equipments for collection of this semen. A separate artificial vagina is used for each collection. The latex sleeve has to be installed in such a way that there are no folds inside the casing. The sleeve should be moderately stretched in order to pour water and blow air. The water temperature in vagina should be 40 to 42 degrees Celsius, and the temperature of the container should vary between 35 to 37 degrees centigrade. And then most importantly, the inlet of the vagina should be lubricated such as a Vaseline. In collection, ensures that the bull does not touch the rump of the teaser with its penis. The person collecting the semen should grasp the bull's penis by the prepuce and direct it to the inlet of the vagina, and then artificial vagina should never be put over the penis. So here semen collection and artificial vagina procedure. As presented here in the hand of a doctor is collection artificial vagina. Here is the shown artificial vagina method for collection of the semen from the buck. Process of the semen collection. After collection, prevent the semen from sunlight, cold, and dust. And because sperms will die if temperature exceeds less than or greater than 45 degrees centigrade or less than or equal to 37 degrees Celsius. After collection, it is important to evaluate fresh semen quality, which will be discussed in our later video, that what are the parameters to be evaluated before processing. So thank you very much. If you are new here, then it will be requested that kindly subscribe and like our YouTube channel for support. Thank you very much.